Dear students, Namaskar. I'm Dr. Nutan Prakash Vishwakarma, and in this video, I'm going to talk about some brief history of bioinformatics. So, as such, the history of bioinformatics, if you look in the literature, there's no clear-cut timeline from where bioinformatics was started. But we can trace back the history of bioinformatics since the work of the Gregor John Mendel. You all know that the work of Gregor John Mendel was a very good milestone in the biological research. And that research was entirely changed the mindset of the people, and still we use the applications of that Mendelian genetics. Why I'm saying that bioinformatics origin we can trace back from the work of the Mendel? Because before that, before Mendel, actually he did a very peculiar thing. That first time he used to demonstrate the result of biology with the help of mathematics. So that was nothing, that was just the starting of the interdisciplinary sciences. As you know, the bioinformatics is an interdisciplinary science. So that's why we trace back the history of bioinformatics since the work of the Gregor John Mendel, right? So he did a very fantastic work, and first time he explained the biological research with the help of the mathematics. Between the 1865 to 1950, a lot of research were going on in the life sciences. And meanwhile, from 1940 onwards, there was the parallel evolution of computers also. From 1940 to 50 onwards, we say that was the generation of first generation computers, right? So the parallel evolution were going on for the computers as well as in the biology. In 1953, one very big discovery, that was the one very big invention, you can say. In 1953, the double helical structure of DNA was solved by Watson, Crick, and Franklin. After 1953, the major breakthrough what happens in the history of bioinformatics that I'm discussing here. Maybe many things are missing, but the major thing which I understand, or what I think it is important that I'm discussing here so far. So in 1955, the name of scientist was Frederick Sanger. First time he solved the bovine insulin. Insulin, you know, it's a kind of the hormone which is proteinaceous in origin. So first time he solved the primary structure of insulin protein. So he come to know that what are the various amino acids in the insulin and what are the order of it. As you know, in our daily life, if any technology comes, then people slowly starts following it. So the same thing happened with the technology, once technology techniques of the protein sequencing developed by Frederick Sanger was came into existence. Scientists throughout the world, they started doing the protein sequencing. And obviously, if you are in the research, then you know very well, if you want to showcase your research, you want to showcase your finding, you want to tell, or definitely would like to tell to the world, then what will be the first step? What will be the right way? How you can tell to your scientific community that you have done something new? So the best way is definitely you would like to go for the publication. So in 1955, there was no internet, no telecommunication. So you can imagine the only way you can publish your work in your countries, the different journals were publishing. So each of the scientists, they started doing the publication in different journals located throughout the world. So you just imagine. And in 1958, one more very big breakthrough came in the place, that is the central dogma. Central dogma of molecular biology is DNA to RNA and RNA to protein. Means DNA to RNA is transcription and RNA to protein is translation. So that was central dogma was proposed, and that was also proposed by Francis Crick, who was involved in the solving the structure of the DNA. That was the double helical structure. After 1958, in 1961, the genetic codes that that was discovered by Nierenberg and Corona. And parallel, the protein sequencing was done in last 10 years. You imagine yourself that you are in the 1960 and you are also planning to go for the sequencing of a particular protein. What will be your first step? Definitely you would like to know that whatever protein I am targeting for, is it already have been sequenced by someone or it is totally new? In case if you've done the sequencing and you gone through the publication, then you and if someone told that this sequence have been already done by someone else, then whatever your effort you made, that is the wastage. So just I'm giving you the situation. You are in the 1960 and you are planning to go for the sequencing of a particular protein sequence at that particular time. It was quite impossible task to go for the observation of throughout the world's journal that whether that particular sequence has been done or not. That is quite impossible. So in 1965, one lady scientist, the name of the scientist was Margaret Oakley Dayhoff. You can say M.O. Dayhoff, or sometime in the examination it is also said Dayhoff. She was working with one organization. The name of organization was National Biomedical Research Foundation. So what she did, she managed to collect all the journals available at that particular time, and she extracted all the protein sequences published till dated in different research papers. And what she did, she compiled all the protein sequences and she published a single book. And in that particular book, there was a uh, only protein sequence related information was there. So in the 1965, you can see the front page of that particular book. The name of that book was Atlas of Protein Sequence and Structure, and that was first time published in the 1965 by M. Udehaf. Now the perception of the scientists towards the protein sequencing was totally changed. Now rather than searching the protein sequences in different journals, now scientists started searching the sequences in that particular book. All right, and that was so popular, and later that book was digitized. As computer were evolved, there was the programming languages and all, and later on it was digitalized. So that's why this is also known as Atlas of Protein Sequence and Structure, is also called as first database. So the credit for developing first biological database goes to Margaret Dayhoff, and a lot she contributed in the field of bioinformatics. So by just uh, by her work, due to her work only, she is also being now known as founder of bioinformatics or father and mother of bioinformatics. This credit goes to Margaret Dayhoff. In 1970, many things were happened. In 1970, the group of the scientists, the Maxim and Gilbert, they came ahead and first time they introduced the technique for the sequencing technology of nucleotide, means especially the DNA. But since we use a radio leveling method and all, so that's why that technique was not so much popular. But still, that paved the way for the sequencing. One more thing came into existence, that starting from the 1955 to 1970, around 15 years of span, there was a lot of protein sequences. And in fact, in the first edition of the Atlas of Protein Sequence and Structure, there was 65 protein sequence was published by Margaret Dayhoff in that particular book. So now in this 15 years of span, a lot of protein sequences were came. So now scientists were started thinking, they started analyzing that is there any sequence similarity between all these protein sequences. So the scientist named Needleman and Wunsch, they developed an algorithm for the alignment of protein sequences or nucleotide sequences. By using this algorithm, and still we use this Needleman and Wunsch algorithm, by using this algorithm, you can calculate the percentage similarity or identity that two protein sequences or two nucleotide sequences share. All right. In 1970, one more big thing happened. The term bioinformatics was first time used by the lady scientist Pauline Hogweg. You can see in the picture, she is Pauline Hogweg with Ben Hesper. He is Ben Hesper. And he defined bioinformatics as the study of informatic process in biotic system. But her work was not known, not noticed much around 20 years. And later, she was credited that she first time used the term bioinformatics. So in this discussion, we learned two things. The first database was made by Margaret Dehoff, and she was the founder of bioinformatics, and she is also being called as a father and mother of bioinformatics. And term bioinformatics was 5 year, approximate 5 year later of the work of Margaret Dehoff. The term bioinformatics was coined by Pauline Hogweg and Ben Hesper. Let's move ahead. The major breakthrough, what happens in the history of bioinformatics that I'm discussing here. In 1977, to overcome the limitation of the sequencing, nucleotide sequencing, Frederick Sanger again came into picture, and he gave the chain termination method or dideoxy method, due to which now scientists started doing the DNA sequencing frequently. So as I told you earlier, there is the long history. Previously, for the protein sequencing, people started doing the publication in the different journals. Then finally, that sequence was copied and pasted in, put it in the atlas of protein sequence and structure. Here, obviously, at that in 1977, the computer was much advanced. So rather than publishing in the journal, now for the DNA sequence, there is the directly the scientists started doing the digitization of the sequences, and one database came by the USA that was sponsored by USA, launched by USA, the name of the GenBank, and that was the annotated collection of all publicly nucleotide and protein sequences available at that particular time. 2 years later, 1 year later, one more database, the EMBL. The name of the EMBL database was European Molecular Biology Laboratory, but the new name is now European Nucleotide Archive. Now the name have been changed. So that was launched by Germany. They also developed one database, and that was publicly available to the people for the use. After 1980, one more algorithm came, the major algorithm, which is still we use, the Smith-Waterman algorithm, and that we use for local alignment of the sequences. So these are major milestones, breakthrough in the bioinformatics history that we are discussing. After 1981, there was again one major discovery, and there was invention, we can say, by Carey Mullis. That was nothing, the PCR, the polymeric chain reaction. Here, whatever sequences till dated till 1983, we will count through the sequencing and all. There, the application of bioinformatics, you can see that once you have the DNA sequence, you can design the PCR primer. Right? So here you can see that now the benefit of the bioinformatics came. So now with by using the bioinformatics, one can design or synthesize the PCR primers, and you can go for the amplification of the DNA. So after 1983, in 1986, first protein sequence, the digital protein sequence database, that SwissProt, curated protein sequence database, and was strived to provide a high level of annotation, came into existence. In 1990, the well-known project. Here we are discussing the major milestones we have achieved during the course of evolution of the bioinformatics. I'm discussing here. So in 1990, the Human Genome Project was launched by the US government with the goal of determining the base pairs that make up the human DNA and identifying and mapping all of the genes of the human genome which human genome has. So in the starting, the project was the target was that they are going to complete this project in 15 years of a span, but due to technical advancement, the goal was achieved 2 years before. So after 1990, before the completion of the Human Genome Project, the first organism genome was published. That was Haemophilus influenzae, that was a bacteria. Because due to the ethical issue, no one can conduct any kind of the experiment on the human being. But still, scientists have to no, they want to predict the function of any particular gene what they mapped in the human genome project. So there was concept came that is known as comparative genomics, because in the other organism we can do the experiments. So whatever experiment we do on the organism, if we match, we align with the human genome, then we can reduce the function, whatever function of any particular gene of particular organism have. And if that particular fragment or gene is showing the similarity with the human genome, we can impose that function to the human gene also, right? So in 1995, the first genome was published of Haemophilus influenzae. After 1995, one more sequence was published. There was the fruit fly genome. And finally, in 2003, the Human Genome Project was declared complete in April 2003, exactly after the completion of 50 years since the work of DNA as a double helical structure. So the completion of the human genome opened a lot of secrets of our life. And due to that only, a lot of discoveries, a lot of invention takes place. And a lot of, since the parallel evolution of sequencing technologies also, a lot of technology came like first generation sequencing, second generation, next generation sequencing. So now the sequencing is very cheaper now. And that's why scientists throughout the globe, they started doing the sequencing. So there is the flow of the data, there is the flood of the data, right? So for that one, we have to obviously to manage such a massive biological data, we need some databases. And obviously to maintain the databases, there was a requirement of the professionals who understand the biology and understand the computer science as well, right? So there was the need of some bioinformatics professionals. And that's why after 2003, or before that, also 2-3 years before, the professional courses in bioinformatics were started throughout the globe. So after 2003, 2004, as I told you, that sequencing technologies were improved. That 454 sequencer was launched by Roche, company Roche. 2006, Illumina came into picture and they launched the Illumina sequencer, which was very quickly you can go for the sequencing of any particular genome. So now the flood of the data, biological data is coming up in the picture. A lot of biological data is coming. That's why more number of professionals are required and analyze to manage and analyze the biological data. Because until you don't go for the analysis of the biological data, to gather the biological data and to manage the biological data, the purpose is not going to be solved, right? So in 2008 to 15, since in 2003, whatever genome project was declared, that nobody knows actually, that was totally secret and nobody knows actually the whatever genome USA government launched, that belongs to that genome is from Pakistan or from India, from South Africa, from USA itself. Nobody knows which sequence was actually launched. So to understand better the variation of the human genome, one project was launched, that was launched in 2008. That is known as 1000 Genome Project, and that was completed in 2015. So after 2015, we have more human genomes so that we can understand better way the human genome and the secrets, the mystery hidden in it. So these are the advancement was going on in 2009. India also played a very good Indian scientists also played a very good role. And first time in India, and the one organization is there, that is the CSIR IGB, Institute of Genomics and Integrative Biology, New Delhi. First time they sequenced the human genome, the Indian genome on the 8th December 2009. And government recently, the IGIB also completed the indigen project, indigen programs. Actually, in that one, they sequence 1000 genome. This is also known as 1000 genome project of India. So in that one, they are going to they have done the sequencing of more than 1,000 Indian peoples so that we can understand the genetic variation and all, the rare genetic diseases and all in a better way as per the Indian population is concerned. So this is the brief timeline in bioinformatics what have been done till date. So I hope you got the better idea about the historical aspect of the bioinformatics in Indian scenario. I hope you like the video and you like the content also. Please like, share and subscribe. See you soon in next tutorial with some other topic. Till then, take care. Thank you.